So hi everyone and welcome to the last session of today's MyTutor online school. This session is GCSE Biology and we're looking at photosynthesis. This is the second photosynthesis lesson that we're doing. We had another one on Monday. My name's Charlotte. I'm the biology tutor for the online school. So I've tutored everything so far. If you've been to some of my lessons before, then welcome back, which I think is quite a few of you. For those of you that are just joining, hello, you're in the right place. So I have been tutoring with MyTutor for about 3 years now, done quite a few lessons, and I studied conservation and biodiversity at the University of Exeter. So my biology background is mostly in kind of ecology and zoology, but I studied a very broad degree, so it covered most of the areas that we run through at GCSE as well. Okay, so as I said, today is the second lecture on photosynthesis, lesson on photosynthesis. Last time we ran through the photosynthesis equation and we ran through all of the factors that limit the rate of photosynthesis. So we looked at the graphs that show how light intensity, carbon dioxide concentration and temperature affect photosynthesis in plants, and talked through how all of that affects plant growth, as well as the process of photosynthesis itself. So today's lesson will be another 1 hour lesson. We finish at 3. And we're going to run through how plants use glucose from photosynthesis, so all of the different things that they do with glucose and how that contributes to the life of the plant. And then we're also going to talk through how we can manage photosynthesis as well. Okay, so let's run straight on through into content. Oh, I need to tell you about the mini quizzes as well. So mini quizzes today are going to be through Zoom. So sorry if those that got very excited about Kahoot or quizzes, we will do some of that next week. But today we've just got mini quizzes through Zoom. So for those, any answers to those, pop them in the chat. So at the bottom of your screens, you've got the chat button and the Q&A button. So I try and use the Q&A button for questions about the content, and we'll have a few stops through the lesson where I'll answer questions. So anything that you're confused about, or if you want me to go back to something, pop it in there, and I'll try and do that at kind of regular intervals. And like I say, any answers in the chat, and we will do a Slido at the end. Okay, so let's go on to how plants use glucose from photosynthesis. So a little reminder here of the equation for photosynthesis. So carbon dioxide plus water with the addition of light gives glucose and oxygen. Oxygen is very much a byproduct of photosynthesis. It's a very handy byproduct because it provides, well, it's part of what the rest of us, so any kind of animals and a lot of other cells need for respiration. So oxygen is the key component in the air that we breathe. And for plants, obviously plants respire as well as photosynthesize. So having that oxygen byproduct is very handy for that. So plants almost have this kind of cycle of photosynthesis and respiration where they're producing their own food and then they're breaking that food down to produce energy for their growth. So we are going to look at respiration next week. So if you're a bit unsure about respiration and how all of that fits together, don't worry. Next Friday, we have a lesson on respiration. So put that in your diaries. So some of these, these types of molecules should be familiar to you all. Certainly right at the beginning of the online school, we did look at digestion. And we talked about carbohydrates, proteins and lipids and how they are processed in the human body and what they make up. And then glucose is also used for respiration. So those are the kind of four main uses for glucose in a plant, and we're going to talk through them now. Oh, I can see someone just asking about the exam board. So this is based on the AQA exam board. If you're not studying AQA, this is still relevant. So we've kept it quite general. So a few examples and things might be different, but photosynthesis is a topic that comes in every exam board. So don't worry, you're not wasting your time. Okay, so we'll start with how plants use glucose for carbohydrates. And there are two types of molecule that have a key component of glucose in plants. So you can see this diagram in the middle here of the screen with the purple and yellow hexagons. So we have glucose there in the pink, and then they've also given examples of other carbohydrates that are present in plants. So fructose is another one that's used in fruit, funny enough. Sucrose is another type of sugar, so that's two sugars joined together for sucrose. And then a polymer of glucose, so lots and lots of glucose all joined together in a row, is starch or cellulose. So both starch and cellulose are lots of molecules of glucose all joined together. So they're a polymer. Cellulose we'll talk about first. So cellulose is used in plant cell walls. So we've talked about different types of cell, and plant cells have a cell wall. It's what makes them different from animal cells, one of the things that makes them different from animal cells. And it's a really tough cell wall. It keeps the cell in its shape and helps to give plants their kind of rigidity in how they stand up things. So cellulose is quite a tough carbohydrate. It's present in all plant cells. So really important. Glucose is a huge part. Well, it's all of that. The second carbohydrate form of glucose is starch. So starch, again, is a long line of glucose molecules. Starch and cellulose are slightly different. They have other components, but you don't need to know them in detail. You do need to know that both of them are formed from lots of glucose molecules. So starch is the way that glucose is stored. So starch is kind of like the larder in plants. So glucose by itself is soluble. It's small enough that it can dissolve in water, and so it can pass in and out of cells. We talked about osmosis. So glucose is part of what can be dissolved in water, and so can pass between cells by diffusion. So starch, storing glucose as starch prevents that soluble glucose affecting osmosis and the water balance in the cell. So glucose, as we've just said, can pass through cell membranes by diffusion, but in doing that, because it's dissolved in water, it affects the solubility of the water, or the amount of stuff that's dissolved in the water. And we talked about different types of solutions, so isotonic solution, hypertonic and hypotonic solutions, and how that affects osmosis. And obviously, if you've got lots of glucose in your cell, it means that the water solution, I suppose, inside the cell is very concentrated, i.e. there's lots of things dissolved in the water inside the cell. And if any water outside the cell is less concentrated, then it will flow in. So it'll be hypotonic to the cell, and it will flow into the cell, which for plants they quite like, but only up to a point. So starch is a way of storing that glucose so that it doesn't affect that water balance. That starch, which is usually stored in leaves, it can then provide energy when it's dark. So obviously photosynthesis is happening during the day and producing glucose then, but at night when there's no light, photosynthesis stops, but the plant needs to keep respiring because it needs to keep producing energy from that glucose. So starch allows that glucose to be stored, and then it can be broken down into separate glucose molecules at night and used then. So sometimes starch is stored not in the leaves, but as tubers for winter storage. So if plants reduce all the way down in winter and don't have very many leaves, or if you're somewhere where there's not very much light in winter, then having a tuber full of glucose in the form of starch is a really good energy store to get you through the winter. Those tubers are often in the form of things that we eat, because we need glucose too, and we get that through our food. So potatoes are a great example of this. Onions are another example. Beetroot, swede, carrots, any of those are storages of starch by the plant, and things that we can eat in order to get our own energy. So that's carbohydrates. Oh yes, you need to know how to test a leaf for starch. So this is quite a nice practical that you either will have done or are going to do. So it's very simple. You might have done tests before for starch. You use iodine for starch. But when you look at plants, you do it specifically for a leaf to see whether it's storing any glucose as starch. So to start with, you boil the leaf in ethanol, which destroys that waxy cuticle on the top of the leaf. That allows iodine to enter the leaf. And you also want to remove colour from chlorophyll, which that ethanol does as well. You want to remove colour because it's a colour change that you're looking for, so you don't want that to be contaminated by green from the chlorophyll. Then you rinse it in hot water to soften the leaf down, add iodine, and in the presence of starch, that iodine turns blue-black. So it's a very simple test: ethanol, hot water, add the iodine. If it turns blue-black, there's lots of starch in there. But you do need to know the reasons behind each step. So for the ethanol, destroying the waxy cuticle and removing the colour from the chlorophyll. Then the hot water is for softening, and the iodine is that test for starch. And there's quite a nice diagram there to remember the 3 stages. Okay, proteins was the next one that we were going to look at. So, glucose and nitrates from the soil are used together to produce amino acids. Those amino acids are what are built into proteins, including enzymes. So, nice diagram here if you're not quite sure about some of the biochemistry of amino acids. You get lots of different types of amino acid, and they can join together to form what would be called a polypeptide chain, which is a chain of amino acids, and they form proteins. So proteins are long chains of amino acids. And the particular sequence of amino acids determines the protein's shape and therefore function. So remember, for proteins, shape is everything, particularly for enzymes. Because enzymes work on that lock and key theory where they are constantly looking for a substrate that fits in that active site, it's complementary to the active site. The shape is everything. So that sequence of amino acids is what, and the bonds between them is what forms that protein into that particular shape. Glucose and nitrates are essential for this. Glucose from respiration, nitrates from the soil. And we talked briefly about how nitrates are taken into root hair cells by active transport when we talked about transport in cells. So if plants can't get nitrates from the soil, they can't make their proteins unless they've adapted to get their protein from another source. So here I've got a nice example there of a bee being eaten by a Venus flytrap. Not eaten, I suppose. But this is another source of proteins, and there's quite a few different plant species that have adapted to do this. So they leave, in this case, what would be their jaws, but their leaves open, with some kind of smell or colour that attracts an insect to it, and then lo and behold the insect lands, and that movement and that pressure triggers a reaction in the leaf, which then closes, and you can see this leaf has got spines which kind of keep that insect trapped. In this case that looks like a bee or a wasp. And then the plant secretes digestive enzymes which basically digest the insect there, and they can then use the protein from that insect to build their own proteins in turn. So that's one adaptation if you can't get any nitrate from the soil. Okay, and we have lipids as well. So lipids, oh, I've spelt from there. So glucose from photosynthesis and energy from respiration are both used to build up fats and oils. Lipids are, well, fats and oils are made up of lipids, and lipids are made up of this glycerol molecule and then 3 fatty acid chains. So glucose is kind of a founding component of glycerol and of some fatty acid chains. So sometimes the glucose is broken down and the different parts are used for fatty acid chains, so the carbon, hydrogen molecules. So glucose is kind of a founder of fatty acid chains like glycerol, and so for lipids. Lipids in plants, so for us, lipids are kind of like a storage of a lot of energy. For plants, they're slightly different. So for plants, their lipids are used to strengthen cell membranes. So cell membranes are basically a long kind of chain of lipids all joined together with several proteins in there as well. So we need lipids to build up that cell membrane. They're arranged as a phospholipid bilayer. For those of you that are going to go on and do A-level biology, you talk a lot about phospholipid bilayers. As well as being used to strengthen cell membranes and organelle membranes as well, lipids are everywhere when you look into different cellular structures. Lipids are also used for seeds. So they're a really good source of energy for a seed when it's starting to germinate. So inside the seed, there's often a real kind of fatty, oily store of energy. And then when the seed starts to germinate, it's got that ready source of energy, which is stored as lipids. And that allows them to kind of get that bit of head start, a bit like an egg yolk would be for, say, a bird. That yolk is there to provide them that energy to give them a start before they can get their own. So while the young plant is putting out roots and starting to build up a stem and then the first leaves, that lipid energy can provide the basis for respiration. Okay, and here's a nice kind of summary of all of that with a couple of extras thrown in. So respiration we're going to do next week, which is energy release. Nitrates from the soil they've included here, combined with glucose to make amino acid, which are then useful for proteins. Proteins, we didn't really talk about kind of what happens when you have a protein. So as well as enzymes, growth and repair proteins are for building new things. We talked briefly about sucrose as a carbohydrate that's used in fruits. Fats and oils for storage in cell membranes, talked about those. Starch, again for storage. Cellulose for cell walls, we covered those. And finally, nucleic acids, which are covered in some specifications and not others. So nucleic acids are what make up DNA. And again, the glucose or parts of glucose can be used to form those too. So that's one extra there. Okay, mini quiz. So we'll do this mini quiz and then I'll go through some questions. So, have your chat function up question 1, you need to type A, B or C. Which product of glucose helps strengthen cell walls? Starch, cellulose or fructose? Plant cell walls. Type A, B or C. Okay, a few people with the right answers there. Any more answers? Well done those who typed so far, everyone's got it right. Z, Saya, Taib, Jessica, Sarabi. Any more? Any more guesses? A, B or C? Starch, cellulose or fructose for plant cell walls? Okay. Very good. B is correct. It's cellulose that's used in cell walls. Remember not to get that confused with lipids, which are used in cell membranes. Okay, question 2. What else apart from glucose is needed to produce amino acids in plants? So A is lipids, B is nitrates, or C is magnesium. Type A, B or C in the chat. Again, well done. Those so far. All right so far. Well done. Nice. Okay, quite a few there. Yes, it is nitrate. Well done. So nitrate's needed from the soil and then glucose from photosynthesis, and they can come together to make those amino acids. And finally, question 3. So lipids are needed for energy sources in seeds and also for what else? So A is cell walls, B for cell membranes, or C for proteins. Type A, B or C in the chat. Okay, nice. A few different answers for this one. A there, B, with a C. Okay, so lipids are also needed for cell membranes. So B was the correct answer there. Well done those that got that one right. Jessica, Kasaya, Austin there as well. Well done. So lipids, as well as providing the energy, a bit like an egg yolk in the seeds, they're also needed for cell membranes. So remember, cell membranes are different from cell walls, and cell membranes are made up of lipids. Okay, well done everyone. I'm going to go back to the proteins slide. Sorry, I missed that. Someone was looking for the protein slide. Here we go. There for that. I'm going to give you a couple of seconds to pop any more questions in the question and answer session. Okay, I'll just give you a couple of minutes to do that. Eye on those there. Few coming in now, which is good. Okay, I'll answer some of those. Okay, so can plants be tired when they receive sunlight every time in a time of photosynthesis? Sorry about that, I don't know whether you heard the noise. Okay, so plants don't really get tired as such. it does use, so the energy that they use for photosynthesis comes from the light that they absorb. So they're not really using any of their own energy in order to photosynthesize. Respiration uses some energy, but plants don't as such get tired from photosynthesis in particular. Is glycerol made from glucose? Yes. Yes, essentially it is made from glucose. It's a bit more complicated than that, but from GCSE you just need to know that glycerol is derived from glucose. Okay, a few people saying, can I go back to slide 3? No problem. Here we go. I hope you can see all of that. You're not missing anything at the top there. Will a Venus flytrap try to get protein from a human if it was in there? Yeah, probably. I mean, you'd have to be in there quite a long time. But yeah, I mean, say if, I don't know, you managed to sort of slightly injure yourself and a bit of your skin would it ended up in there, then yes, it would digest that. But Venus flytraps are usually small, so you know, if you were somewhere where they were native, you wouldn't get kind of snapped up or anything. But yes, I don't think they're very fussy. It's a good question. I don't think they're specific to any particular type of protein. Someone else saying back to number 7. I'll give a couple of seconds on this one while I answer the next one, and then I will go to slide 7. Is fatty acid just fat? Yes, essentially. They're called fatty acids. That's their kind of the name for that particular part of a fat. So a lipid is made up of glycerol and 3 fatty acid chains. And again, you'll do a bit more into the biochemistry of this in A-level if you do go on to A-level biology. But part of those chains has a little acid group on the end of it. So an oxygen and a hydrogen, I think, is an acid group. So it has those on there as well. Okay, let's go to slide 7. A few people are asking about the mini quiz as well. So the answer for question 3 in the mini quiz was lipids for cell membranes, which I think was B. Let me check real quick. Sorry, guys. Yes, so lipids are needed for cell membranes, so B. And a couple of people asking who got the most right. Let's have a look. As far as I can see, Saya had them all right. I think Jessica did as well, and Sarabi. Saima nearly. I should answer the last one from what I can see. Jessica as well. There's quite a few of you got them all right. Nice one. Okay. Request for science jokes at the end. I've kind of stayed away from them actually. But we can try them out. That's fine, no problem. There's about 30 of us in total in this webinar. A few people asking about those. Okay, Hopefully you've all got everything that you need from the slides now, so I'm going to move on to the next bit of content. Okay. So there was the mini quiz. So the next part of photosynthesis is working out how we can make the most of it as farmers. So how, as humans, can we make the most of, oh, someone else came out, slide 5, be quick. And then we can move on. So last session we looked at how photosynthesis is affected by temperature, carbon dioxide concentration, and temperature, carbon, and light intensity. So this time we're going to have a think about how we can manipulate those so that we can manipulate plant growth. Really handy when you're growing any sort of crop, so particularly vegetables or crops out in fields or in labs as well. Okay, so how can we use and make the most of photosynthesis as farmers? So greenhouses are where this mostly comes into play. So growing plants in greenhouses means we can control all the limiting factors of photosynthesis. We can control light intensity, temperature, and carbon dioxide concentration. This means that we can really affect the yield of a crop. So the yield of vegetables, that might be tomatoes, it might be lettuce, it might be anything else that you would grow in a greenhouse. If you are growing plants for decorative purposes, then you can increase their growth so you can get them ready to sell faster if you're manipulating light intensity, temperature, and carbon dioxide. It's important to remember that these factors interact. So for example, in the early morning, while it's still fairly cool, both temperature and light are low, and so they're affecting or they're limiting how much photosynthesis can take place. Later in the day, when temperature is warmer, particularly at the moment, it's so lovely and sunny, and there's no difficulty there with temperature or light, then it's probably carbon dioxide concentration that might be limiting the growth there, because there's not really much we can do about the amount of carbon dioxide that's in the air. If you look at this graph, this is one that you might see quite often. It's a combination of all three of those graphs that we looked at before, or all three of the factors there. So it's quite difficult to read. And I've realised that it's come out slightly blurry as well, so I will read it through to you here. So on the y-axis, we have rate of photosynthesis, like we did on those last graphs. So this is how fast photosynthesis is taking place. And then on the x-axis, we have light intensity. So they've put light intensity at the bottom, and then the different lines on the graph show how that rate of photosynthesis is affected by light intensity in combination with other factors. So we can see that at the beginning, light intensity increases, or rate of photosynthesis increases with light intensity, along with any other factor that you've introduced. But then it plateaus off in the first place when you have a low concentration of CO2 and a low temperature. That keeps the rate of photosynthesis quite low. And here it's put on the right here that at this point photosynthesis is limited mainly by CO2, obviously temperature as well. If we follow up again, if we then have, so if we change that CO2 and go, okay, it's limited by CO2, let's put more of that in. So now we have a high concentration of CO2, but we've still got a low temperature. You can see that the graph plateaus off again. We've got a kind of middling rate of photosynthesis. It's not too bad, but it's still limited by temperature, as it says on the right-hand side. So if we alter that temperature, make sure that we've got a high temperature and a high concentration of CO2, then we're really flying. So rate of photosynthesis still plateaus off, but at this point, it's limited by something else. So we've got good light intensity, we've got good CO2 and good temperature. We've manipulated all of those to be the best they possibly can be. Now it's just down to the rate that the enzymes can work in the plant. And it's like any activity, you can only go as fast as you can go. It's like packing things in a factory. You can only produce as much as the packers can pack. So as fast as your enzymes can work is as fast as you can go. And you've already provided them with a high temperature. So you've provided them with an optimum temperature. And then you're literally just limited by speed of reaction, I suppose. So that's that graph. It's really good to be able to explain that. So it's well worth kind of writing that one down and maybe writing out a few times how you would explain that graph. So we mentioned before that greenhouses, oh, I've just glanced at the chat and someone has put the golden word, chlorophyll does also limit. So obviously your enzymes can only work so hard and the chlorophyll can only, you can only have so much photosynthesis taking place within the chlorophyll. So yes, you're exactly right. The amount of chlorophyll in the plant is also a limit in there. But there's not much you can do about that. It can't be manipulated really. Okay, so we talked about greenhouses being an ideal kind of habitat, I suppose, for controlling all of those factors. So in really big food production systems, like the ones in the pictures here, really big factories producing mainly vegetables, you can use computer systems to affect, right. So you can use computer systems to control temperature, light, and CO2. So some greenhouses grow plants in water as well with a perfect balance of nutrients, and that's called hydroponics. So you can see in the greenhouses at the bottom here, there's not really much soil. And that's because they're using this system of hydroponics where you have water which just has an ideal balance of nutrients. So you don't have any of the mess of soil. And in that way, greenhouses are a bit like a laboratory when they're this big and this sophisticated. So it allows you to control conditions in a really complicated and very expensive way, but in quite a precise way. So we've got pros and cons here. And this is the sort of thing where you might get a question in an exam saying, evaluate why someone would choose to produce their vegetables in a greenhouse rather than outside in a field. The pros of having really controlled conditions in a greenhouse is that you get a higher yield, faster turnover. You can make your plants grow really quick, so you can harvest them quicker, get them out and sold quicker. Clean crops because there's no soil. So pick them straight out, no washing required, no cutting off of any part that's been soiled by the soil. They can be grown where land is poor because you don't need soil, you don't need that environment, and you don't need that many staff because computers are regulating a lot of those conditions. Cons are that those kind of greenhouses need a lot of planning and a lot of expensive equipment. You need to have a really well thought out and well tested approach to growing the particular crop that you're doing, in this case, lettuce. And it uses lots of electricity and gas, so it doesn't come without its disadvantages. Very expensive and not necessarily that great for the environment because obviously gas has a CO2 output. So lots to think about there. There's all sorts of questions that can come from this. Is it right to be growing our food in this way? It's quite good. You can feed a lot of people quite quickly. It's good, particularly in areas where you can't produce much food. We have a very high population on this planet and food security is a problem. So solutions like this are quite innovative. They don't involve as much space as it would if you planted them out in the field. And so they can be quite good for that. But there's also an element there of this being quite forced production. Like I said, it uses quite a lot of electricity and gas, which are often from non-renewable resources. So there's all sorts to think about there. Okay. So that is kind of the end of the content. That was a bit of a food for thought question at the end there, if you excuse the pun. Okay. Let's run through any questions. So I'll give you a second to answer some questions because normally we would have a mini quiz, but we're going to do a Slido in a minute. So let's do questions first. Write them in the Q&A. I'll just whip back. Someone's asking for slide 5 and 6. Do 5 for a few seconds and then 6 as well. Someone's saying, what's carbohydrates? Carbohydrates is an enzyme that breaks down carbohydrates. Although it's spelt differently to how you spelt it in the question. But I assume that that's what you've meant. Okay. So I'm going to move on to slide 6. Whoever needed the new slides there. Someone's asked, is the effect of using non-renewable sources of energy, global warming, greenhouse effect, cancelled out by the photosynthesis carried out by the plants? That's a really good question. My answer to that is that because it's such a controlled environment, the photosynthesis taking place in the plants, so the product of that we're interested in this case, which would be oxygen, isn't expelled outside. It's kept within the greenhouse because it's such a controlled environment. So in this case, no. But I really like that idea that although you're using a bit more to grow, in theory you're putting more oxygen back into the atmosphere, taking out more CO2 through the photosynthesis, and so helping to mitigate that greenhouse gas effect. And to be honest, there might be ways of getting around that. I mean, if you opened up the greenhouse for a certain amount of hours per day, would that change that? I don't know. So there's all sorts of thoughts there. And I know that, so certainly because I work with farmers now and look a lot at kind of how we can make agriculture more sustainable and different things like that. And this comes right under that bracket. Is this a really good way of producing food without taking too many resources from our planet? And there's lots of pros and cons, and there's lots of people, doing really good research into this now. So if you're interested, there's lots of jobs available. What's the difference between fatty acids and glycerol? So essentially they're made up of in a different chemical formulae. So they're arranged differently. Fatty acids are arranged in a chain. Glycerol is, I can't remember what shape glycerol takes. So they have a different chemical composition. Fatty acids, as I say, have that little acid group on them as well. Again, this is something that you tend to go into more with A-level biology, the kind of biochemistry behind it. So that's possibly not the best answer to your question. But there is a difference between them in their shape and their chemical makeup. Someone's asked if I'm studying medicine. I'm not. I finished studying, so I did study conservation and biodiversity for a master's degree, and then now I work, like I said, with trying to make agriculture more sustainable. What does mitigate mean? So mitigate is where you try to reduce the effect of something, so it's used most often in the context for me for ecology. So if you build a big housing development and you take away some habitat, if you then build habitat elsewhere, you're mitigating for that lost habitat. So you're making up for that lost habitat. Good thinking there. Someone suggested opening the roof of the greenhouse, and someone else has said, oh, but that would reduce the temperature. Exactly. It's really difficult to work out how we can do this and still keep everything at the optimum rate that the plants need. And so perhaps you might think about doing something different at night when the plant isn't photosynthesizing. That would be the first thing I would try to think through. Do I think the coronavirus is a plandemic or made in a lab? That's a very good question. My answer is that I don't think so, but that's a personal answer. I always try to think quite scientifically about these things, and a lot of the health scientists are giving information out that this virus jumped over from bats. But I don't think anyone's sure. And that doesn't mean that any bat that you meet has coronavirus. That's not the same at all. The virus mutated in a particular place and then spread from there. So the bats that you sometimes see flying around at night don't have coronavirus in general. Good, loads of people questioning about coronavirus. It's really good to question. That's exactly what science is about. It's about questioning everything that you hear. And that's exactly why we have peer review. Here as well. Oh, the camera's just swapped. Hoping that's okay. Okay, I think we're okay to carry on. Calvin cycle is part of respiration. But again, you learn more about that at A level. Okay. So I think that's everyone's questions answered. Oh, let's do that Slido before I forget. Pop this in here, and then I'm going to do a word cloud. What are the keywords of photosynthesis? That's open. Now, oh, I can't remember how I share it to you from here. So you can, if you've got your phones, you can use the QR code here, which should be fine. Otherwise, normally share a link. So you can go to slido.com and type in hash E945 as well. So if I put in, good, loads of people in there already. Slido.com, I've popped it in the chat. Loads of keywords coming in, well done everyone. So this is quite different to Monday, where we had lots and lots of keywords, but we didn't have quite so much about the different factors that affect photosynthesis. We had lots of chlorophyll, which again has come up here, and we had glucose, and we had lots of other words. And this is much more thinking about energy, thinking about light intensity, carbon dioxide concentration, and temperature. All of those are in there, which is great. Good. Plants iodine, yes, exactly. Remember that test. Starch, cellulose are both in there. What other things can we make? So apart from carbohydrates, we also have, I'm frantically looking through these, ethanol's in there as well. Good. Fats, yes, that's what I was looking for. Fats and oils, yes. What's the other one that's missing? Nitrates, well done. Now, what's the one linked to nitrates? Nucleic acids are in there, well done. Protein, yes, that's what I was looking for. Proteins as well, well done, everyone. Great. I'm going to take a print screen of this. Use it next time. Couple more seconds. Still got intensity as the most common word there, light intensity. Well done, everyone. Okay, I'm going to stop that one there. Take me print screen. And then we've just got 5 minutes left, so we'll do a quick question as well. Okay, so this is a very simple question just to start off. If you want to do this in the chat, then you can. Complete the equation, draw a ring around each correct answer. Carbon dioxide plus what? With light energy, it gives what plus oxygen? Okay, nice. A few people popping in their different answers. Water and glucose is correct. Well done, everyone that put water and glucose. There's lots and lots of you. Jessica, Austin, Kasaya. Oh, lots of anonymous people. Great, Danielle as well, well done. Okay. Here's a question about an experiment. So some students investigated the effect of light intensity on the rate of photosynthesis in pondweed. The diagram shows the apparatus that was used. So we have thermometer there. Got a glass test tube with pondweed in it. Water is at 20 degrees C, which is shown on the thermometer, and then you've got a lamp moving at a distance from that beaker. So we kind of talked about something similar on Monday. So this is a classic case of investigating how light intensity affects the pondweed and how it photosynthesizes. And that's exactly what they've said here. So the closer the lamp is to the pondweed, the more light the pondweed receives. Students placed the lamp at different distances from the pondweed, and they counted the number of bubbles of gas released from the pondweed in 1 minute for each distance. So that's a really nice way of measuring the rate of photosynthesis, because you can see the oxygen gas being given off. And that's one of the huge advantages of using a water plant. So a thermometer was placed in the glass beaker. There's a birthday celebration going on next door. Why was it important to use a thermometer in this investigation? So pop your answers in the chat. Good. Good. Quite a few people in there. Yes, exactly. So the role of the thermometer is to measure temperature to check that it isn't changing, as temperature may also affect the rate of reaction, so it needs to be controlled. Loads of people have put that in the chat, so well done, Kasaya, Jessica, Pranav, Javita. Oh, sorry, Javita, you put in a question as well. When do you use the question 1 over distance squared in biology? That's a really good question. I don't know whether you do. be honest. I'd have to look that one up. Good. Sarabi is saying that check that it doesn't change. Well done, everyone. Okay. Students counted the bubbles 4 times at each distance and calculated the correct mean value of their results. The table shows the student results. Quick question here. They're saying calculate the mean number, or a maths question rather, a quick maths question. Calculate the mean number of bubbles released per minute when the lamp was 40 centimetres from the pond. So they're asking you to complete the table. Have a good look at the table here. This is exactly the question where you need to look closely and make sure that you're reading it through. So we've got distance in centimetres down here, number of bubbles per minute here. So first minute, second minute, third minute, fourth minute, and then the mean. Okay. Few people answering. Welcome to use nice. Someone popped in 32 there. Another person saying 15, 10. 10's another question. Another answer, possible answer. I'll pop it up. Okay, those of you who put 10, well done. This is a really, yes, so loads of people popping 15 in. 15 would be correct if we didn't have an anomalous value. So this value here is vastly different from the other 3. So we think that it's an anomalous value, and this is why this question is 2 marks rather than 1 mark. So normally work out the mean would be 1 mark, add everything together and divide it by how many there are. But for this one, you had to notice that anomaly. And that's a really good kind of lesson for learning to read a table properly, and make sure that you don't lose that 1 mark, because that 1 mark is always worth having. So for this one, 10 plus 9 plus 11 divided by 3, because you're only using these 3 values. Okay. That is all we have time for, unfortunately. We're finishing a couple of minutes late because we started a couple of minutes late. So I think we're going to have to stop there. If you want to look up more photosynthesis questions to test your knowledge, then I can highly recommend using BBC Bite Size, or Physics and Maths Tutor is another really good website where you can look up questions by topic, which is great. If you'd like any one-to-one support, remember that MyTutor do one-to-one tutoring as well, so have a look for that on the website. And keep your eyes peeled on the timetable for next week. I think next week's lesson is on Friday at 2 o'clock again, and we're going to be doing respiration. Day off on Monday, 3-day weekend, so that will be nice. And I hope it's sunny for you all and you'll have great fun plans. Few more questions there. Oh no, a science joke. Thanks for making me laugh, Grace. Okay, all right, have lovely weekends, and I hope I'll see you all next week. Bye, everyone.